objective is to take care of mitochondria, right? We're not going to look at the overview, what mitochondria is, the anatomy. I'm not going to stand too much on that because we already know what that is. And then we're going to talk about the mitochondrial dysfunction, as well as the cell danger response and responding to the environmental and then chronic illness, encompassing the inflammation, innate immunity, and oxidative stress and stress response. And I'm going to have a case study I'm going to present at the end as well. So in dealing with the mitochondrial anatomy, mitochondria is located within all cells, largest concentration within the skeletal muscle, brain, and kidney, has its own genome and DNA, and we already know this, right? It's responsible to producing ATP to fuel the cells, organs, muscles, and the brain. And the mitochondrial structure actually has two different membranes composed of phospholipid layers, and this is very important, which I'm going to be touching about in the later part of it, is that they have a different function within the composition of the membrane, and there are outer membrane, intermembrane space, inner membrane, and the matrix. And when we talk about mitochondria, we cannot not talk about Krebs cycle. Right? We end up having to cringe a lot when you looked at this. And one of the things that Charlene ended up having to mention today was that we're going to have a quiz on this diagram at the end, so make sure you copy it down, and we're going to do a Q&A and as well as a quiz on this, all joking aside. But this is the things that we end up having to cringe about during our biochemistry class in college, right? And now I'm actually able to appreciate a lot more the fact that how these are all intricate in terms of allowing us to know about the overall functional medicine approach in terms of patient's health, recovery aspect of it, any micronutrients you might be needing. And then one of the key things that we are having to appreciate about this entire cycle is to do what? To produce energy, right? So mitochondria synthesizes the adenosine triphosphate from adenosine diphosphate to allow something called the oxidative phosphorylation. We know this, right? Nothing new. And then there's about 90% of the energy are produced within the aerobic metabolism. And the entirety of the citric cycle is that it's able to break down pyruvate from glucose and allow fatty acid to be utilized and broken down. So nothing new, nothing to really talk full about. But one of the things that the Krebs cycle and allowing us to really work on within the electron transport chain, 90%, again, 90% of the ATP is produced, and rightfully so, right? Is a heavily well-efficient running machine. It's just that when you're dealing with chronic illness, stress, I know Dr. Horace actually talked about Lyme disease this morning. You know, a lot of these things end up having to come into play to make the mitochondria in a dysfunctional state, right? And certain nutrients also really affect how this particular electron transport chain ends up having to work. CoQ10, for instance, is a high energy electron transfer between various mitochondrial complex. And then when you talk about carnitine actually helps to transport that particular CoQ10 within this different matrix and energy as well. And ribose, which is one of the key things that we end up having to support when you're talking about cardiovascular health, right, is a backbone of ADP and ATP. So mitochondria function-wise and protection, it is very susceptible to nutritional deficiency and mitochondrial toxins and oxidative stress. It also determines, based on how much of a damage that the mitochondria incurs, it determines how the overall ETC is going to function. And so just to give you an example, we end up having to, my pharmacy, for instance, any pharmacist out there right now, we dispense a lot of statin drugs, right? Statin drug itself is widely used within medicine. However, it does end up having to deplete CoQ10, it depletes vitamin K2, but when you look at this aspect of it, dose dependency, depletion of CoQ10 in a patient taking statin drugs, and is resulting in an oxidative stress in cell leading into leaking of oxygen. And so cell function and mitochondrial function is not an efficient matter when you're adding in statin drugs on top of everything else. So we got to think about how the drug is being used, and we need to also, particularly, I hate doing it a lot of times, to educate patients about certain drugs that could potentially cause mitochondrial damage, right? Acetaminophen, not surprising, right? It actually depletes glutathione. Amphetamines, we dispense a lot of Adderalls these days. Kids actually having ADD left and right. Is it more mitochondrial dysfunction that's causing it? Is it a food sensitivity? Maybe both, right? And then when you're talking about certain NSAIDs and then PPIs, which are also widely used again, we have a lot of dysfunctional state within the mitochondria we're promoting without even thinking about this. And then when you're adding in the antibiotic portion of it, it's going to wipe out the gut microbiome, right? In itself, when you're using it chronically a lot of times, it can also damage the cell membrane of the cell itself, as well as causing the overall mitochondria in a dysfunctional state. So the role of the mitochondria is to synthesize ATP. It involves in a calcium homeostasis, promotes cell growth and cell signaling as well. Apoptosis, very important in dealing with cell danger response and particularly leading into certain danger and the damaged cells to be renewed, and generating other oxidative radicals, and then supporting nerve conductivity as well. In additional roles, cellular synthesis, hormone signaling, and as well as, again, cellular apoptosis and programmed cell death. Now, shifting into what's causing the mitochondrial damage, we talked about drugs before, right? Aging is probably number one. Accumulation of oxidative damage, not surprising, right? Genetic susceptibility could be one of the issues. But gene, you got to remember, is just a loaded bullet in a gun, right? In itself, how it gets triggered is based on the person's epigenetic profile in terms of how the person's lifestyle is, what they eat, how they sleep, how they poop, right? How they deal with the stress issue. A lot of these things can actually cause our genes to be expressed in a different way. So there's no point to be worried about as long as we're able to take the actions possible to help protect us from the issues that might be from our genes. There are toxic metals you have to deal with, right? We talked about toxic metals just before, how, total tox panel. I love using them, actually, in my patients, and they're able to utilize that to really determine what type of environmental toxins and heavy metals that come into play, right? Persistent organic pollutants, like glyphosates, is one of them, and then as well as other preservatives, as well as other chemical pollutants out there. Many prescription drugs, right? All joking aside, friends don't let friends take drugs, right? Drug in itself, I dispense it. We take care of patients all the time, but it's not very optimal in terms of taking care of the mitochondrial dysfunction the patient might be dealing with. And then you have alcohol, you have mold parasites, Lyme disease, and a slew of other vector-borne illness issues as well. So strategies-wise, how do we strategize in terms of improving mitochondrial function? Well, first thing, I hate to say this, but you need to pick the right mother. As a matter of fact, I would include the father in there as well. Why is that? It's because you need to have an optimal mitochondrial DNA for better offspring, right? So one of the things I encourage any families that are planning actually having a family for that matter, they should think about prepping for at least a year or so to detox, to get their hormone optimized, especially the nutrients as well, to allow themselves to have a better health, to have better offspring, right? This also in turn, I was at a CellCore conference last month, and actually back in August, and one of the lecture was just talking about how do we take care of other biotoxin burdens to allow healthier pregnancies. This should be talked about, right? Because we're dealing with a cesspool within the environment, right? Chemical toxicity going on. Many things that we need to take care of before we're able to have a healthier baby as an offspring. Next, optimizing nutrient status. Not surprising again. Exercise is a big deal, right? Building muscles, having an adequate muscle mass will add a lot of dividends within your health, right? Even with the mitochondrial damage from Parkinson's disease, having a 15 minutes of activity can actually help with the mitochondria to produce energy. Sleeping is most important as well, right? Recommending about 7 to 9 hours sleep. You want to block as much of EMF as possible, right? If you have an EMF blocker, or for me, for instance, I started using this grounding bed mattress mat, and it's wonderful to block off unwanted EMF. I didn't realize how sensitive I was with EMF, and just using that alone helped a lot. Well, that's going to help the mitochondrial function, right? You want to use a blue light blocker, and then possibly optimize an environment for you to get a better sleep. Next, you want to limit the sugar, okay? Because why? It actually impairs mitochondrial energy production. You want to decrease toxin exposure and getting rid of BPA. When you go to a supermarket, for instance, and they give you a receipt, How many of you actually take those receipts and take it home? I don't, not anymore. Why? Because it's laced with BPA, right? And then when you're talking about those workers end up having to take alcohol to sanitize their hand and then having to grab into BPA, what are you doing? You're absorbing all that toxin into your body without even realizing it. And so if you can, ask them to email your receipt instead of taking it home. Better yet, don't even ask for it in many cases. Next, you want to provide nutrients and protections for the mitochondria from oxidative stress. One thing that I started doing about 2 years ago during the pandemic was using far infrared sauna. I don't know if you guys are familiar with the company called TheraStage. I use it every single morning. 20 minutes, 170 degrees. I sit in there, sweat it out. What it's doing, it actually helps to empower the heat shock protein to allow your immune system to work optimally. At the same time, it actually helps to induce mitochondrial repair. Following that, I take a cold shower. I took a cold shower this morning. It was wonderful. I think everybody should be taking a cold shower every morning to wake up. Everybody's laughing because, are you doing it? No? You should do it. It actually helps. And then fasting. 3-day fasting is one, and then fast mimicking diet. This is what I really give to patients. Fast mimicking diet is called Prolon. It was designed by Dr. Walter Longo. Helps with improving autophagy and mitochondrial function and detox. Really helps to reset the metabolism as well for a lot of these patients who are really stuck. Well, this is going to help with the mitochondrial function. Very well put together, so I would highly encourage people to add it into the entire program. And lastly, to minimize pharmaceutical drug exposure. Essential nutrients for ATP production, for beta oxidation, riboflavin, NADH, and carnitine for transporting fatty acids. Where do we get carnitine from? Red meat, right? Animal protein. But the whole entire push of using plant-based food, or at least veganized food, is actually affecting a lot of this particular health and supporting the mitochondria. So I'm not saying to go completely carnivore or anything else, but really have to look at what dietary trend is actually happening at this point. With this whole thing with Impossible Burger, it's not really helping anybody. It actually has inflammatory oil, is really offensive in terms of the body, how it's used. And so I don't really recommend it. And especially patients who are coming with chronic illness, you gotta ask them what kind of food they're eating. So this way we could at least clean up their diet. That's gonna fix a lot of issues to begin with. Next, the citric acid cycle, iron, magnesium, manganese, cysteine to actually help producing glutathione. And then for electron transport chain, CoQ10, NADH, and so on. The last thing I would encourage really doing is adding in the phosphatidylcholine. Phospholipids helps with the cell signaling and also is a resolution aspect in dealing with inflammatory cycle. And this is a very important part I'm going to discuss later on of this lecture, how to respond to the mitochondrial dysfunction and utilizing phospholipids. Nutrient recommendation again, vitamin C, vitamin E, and fish oil-wise, EPA and DHA. Yeah, it does help with the controlling oxidative stress, but I don't really recommend it as much, right? Because there has to be a right balance between omega-6 and omega-3. And giving too much omega-3, it's also not very beneficial for patients' overall immune health either. So you got to do a fine balance, and I would also encourage maybe looking to test the patient's lipid profile, right, and see how their omega-6 and omega-3 balances are, and really adjusting based on those levels. Next, magnesium is not surprising at all. So mitochondrial dysfunction in dealing with early aging, diabetes, hypertension, right, these are all part of mitochondrial dysfunction. And this can be dictated a lot of times based on what type of exposure that we're going to get from certain environment. So shifting our gears and talking about cell danger response, this particular term was coined by Dr. Robert Novier and all the research he has had done. He's based out of UC Davis, and a lot of time things that he had put in together might be theoretical a lot of times, but it's really tried and true testing that he had done in his lab and makes a lot of sense. So cell danger response, or CDR, is an evolutionary metabolic response to protecting cells and hosts from the harm. And they are triggered based on chemical, physical, and biological threats. And biological, viruses, bacteria, mycotoxins, and parasites, chemicals such as heavy metals, and then also dealing with certain electrophilic aromatic chemicals like BPA, right? Flame retardants like brominated flame retardants out there. That's also can be very toxic for your mitochondria. Physical, you have talking about heat, shock, or pH changes, and as well as sun damages. And then psychological traumas as well. We don't really rarely talk about that. This should be one of the things that we should be talking about in screening patients in dealing with potential trauma as well as other psychological effect they end up having to get through. But these are 4 things that could be triggering something called the cell danger response. And what that actually pushes is that the mitochondria alters its ATP production and it shields a further injury. So it goes into overall battle mode, downregulates the protective mechanism that the mitochondria does, and it goes into the entire process of fighting, right? The mitochondria senses that. How? Well, let's take an example of a virus, for instance. If a virus ends up having to be present, it causes a depolarization within the cell, of the cell membrane, which alerts the mitochondria to do what? It starts to decrease the oxygen consumption and goes into blocking and decreasing the RNA proteins and DNA synthesis that the infecting pathogens or insults that can actually occur. So there's many different inflammatory fatty acids, there's acetate forming, as well as one of the key important part of this entire process is that mitochondria end up having to orchestrate this entire process, right? You're like, wait a minute, I thought we were only talking about producing energy. That's not the case at all. Mitochondria detects as well as allows the entire process for the body to go through this thing called the cell danger response. Another thing that the mitochondria also does is it allows the increase of phospholipids to stiffen up this membrane to limit the spread of different pathogens, right? And this is an important part we're going to touch upon again, but this is going to limit other pathogens and other potential toxins to get involved to causing further damage. And this is what the mitochondria does. Next, it releases antiviral, antimicrobial chemicals in there. Detects autophagy. At the same time, there's different changes of DNA methylation. Also, it pumps out reactive oxygen species, aldehydes, and polyamines. Then it also affects the ATP as a particular signal molecule, right? It's not an energy now. ATP turns into a signal molecule to allow other cells to come into play, such as T cells and phagocytes. And another thing that it does is it triggers the host behavior to be changed in that sense of that the CNS gets triggered to sickness behavior, and then the entire process of what the mitochondria is trying to do and the body's trying to do is to contain the damage, allow the healing mechanism to come into play. So this is what the healing cycle looks like, right? There's a different steps of it. There's certain, and by looking at this diagram right now, does not mean that you're going from cell danger response 1 to 2 to 3 in a significant order. A lot of times, some patients may end up having to be stuck in two different modes, or may not be able to get through it at all. So let's to give you an explanation on this. So stress, injury, or toxin infections end up having to cause a innate immunity response to occur within the body, right? So for instance, a virus end up having to be played. Matter of fact, let's talk about COVID-19, right? So that in itself, the innate immunity end up having to come into play. Now what's the next step is to contain the particular damage. And so there's an excessive metabolic changes that goes on to cause apoptosis and necrosis and phagocytosis to occur. And the body is going to what? To contain the inflammation, contain the damage, and to allow mitochondria to dictate all these things. Next, once the CDR1 process is contained and somewhat controlled, next process is going to be on the CDR2, which is the proliferative phase where there's an accumulation of biomasses. So give you an example. Let's just say you have a cut, and the next step is for healing to occur. What happens is you're going to have different fibroblasts as well as tissue regrowth that occurs. That's literally what CDR2 has been doing. And there will be different sets of proliferation that could be damaged in dealing with that, depending on how much of exposure of the damage you're going through, as well as a person's genetic makeup and such. This can actually lead into excessive DNA damage and then senescence, right? So this entire process called the aerobic glycolysis. So Otto Warburg, for instance, he coined this term back in the 1940s, and he ended up having to describe aerobic glycolysis in describing insulin resistance and diabetes, right? The body's not very efficient in producing energy, and matter of fact, it's dumping out energy based on that. And that's where the process of cell danger response 2 occurs. Now, once that goes through, and the next phase is to allow differentiation of the cell itself, right? So there's a constant cell-to-cell communication going on. And when there's a missed healing, or at least the healing cycle end up having to be affected, this is going to cause a different functioning of the cell in itself and change it. And now, this sounds a little complex, but just to go through that, CDR1 is an activation of the innate immunity, and is able to detect the actual intruder, removing it, damage control, and contain the issue, and is the role to decrease apoptosis from exposed to lethal insults. And then once that CDR1 is done, CDR2 is to upregulate aerobic glycolysis to allow biomasses to occur, to accumulate, and allow cells to proliferate. However, if the process is unchecked, it leads to proliferative damages where the cell is not able to communicate that well, and this is where the progression of different disease states come into play, such as autoimmunity, cancer, right? So CDR3, on the bottom of the sentence there, depending on the host genotype and character and developmental timing and the magnitude of the overall damage itself and the frequency of the exposure, end up having to cause a dysfunctional cell danger response to occur, and that's where the chronic disease states come into play. Right? A lot of patients are actually stuck in this matter, right? If you want to go back, right, if you're talking about COVID, for instance, patients dealing with acute respiratory distress, what's happening? They're stuck on CDR1, right? And then when you talk about patients dealing with post-COVID sequela issue, they might be dealing with maybe stuck in CDR1 and CDR2 where they're not able to, let's just say, give an example, gain their overall lung function back, right? They have a 20% less of a capacity to hold oxygen, right? And then they're having other things issue like fatigue, and then having to have other matters in dealing with triggering of Epstein-Barr, or I see a lot of patients now, the reactivation of the Bartonella infection after having COVID. These are all into play in terms of how intricacy of all process that the mitochondria end up having to dictate. Patients are stuck based on this cycle. And so why this is so important is that we don't want to haphazardly get into a case and then throwing some random supplements at the patient, right? We need to allow us to look in a different way in terms of really seeing where the patients are stuck based on the cycle to allow proper healing, but also allow mitochondria to calm and able to allow the mitochondria to orchestrate the entire process of proper healing. And this is, I think, a lot of times is missing because of the fact that we're so reactive instead of actually really looking at the overall case and the presentation, right? So this in itself, let me get a sip of water, is a very interesting diagram of explaining the overall process of functional medicine. When you look at this, you're like, all right, it's a simple chart looking at, healthy development and then innate immunity issues. But if you look at this right now, mitochondria end up having to dictate this entire process, right? It's not just about making ATP. It's not just about cell danger response. Well, in lieu of cell danger response, and as well as a healing cycle, and controlling everything that goes in the cell itself, mitochondria dictates the entire process, right? From using oxygen to making ATP to making glutathione, right? Even the methylation process, for instance, in a healthy methylation process, right? Transfolate and then to methionine to methylation process. But when you're dealing with a particular issue where there's an innate immunity and other stress are getting involved, what's it doing? It actually demethylates, actually making it worse, causing purine synthesis to occur, and DNA damage, right? Even if you look at histamine, for instance, we deal with a lot of patients dealing with mast cell activation, right? We end up having to run all these different labs to see what's causing it, and thinking that we could just give them some medication to be end of that. But if you look at the overall histamine response, it's not surprising. Well, mitochondria dictates that. There's certainly mitochondrial dysfunction that goes on that we have to look at in a bigger way and try to really find the root cause issue, right, in dealing with mast cell activation. Even if you look at vitamin D, for instance, right, vitamin D, anti-inflammatory effect, but in a certainly triggering of the cell danger response, namely because of mold or mycotoxins, for instance, right, it actually causes an increased inactivation of the vitamin D, causing inflammation and possibly autoimmunity. And this is one of the things I end up having to see. And a lot of times I see patients where their vitamin D never goes up because of the progression of the disease itself, right? Even chronic Lyme disease, for instance, I see patients where vitamin D doesn't go up, even though we might be supplementing with maybe 5,000 to 10,000 units. Why? The vitamin D receptor is in a complete dysfunctional state. Why? Because of mitochondrial dysfunction, right? So we have to look in a bigger way rather than just running random labs. And by looking at this right now, right, what are we doing as a functional medicine providers? We order a lot of nutritional panels, right? Because why? We think that there's a nutritional deficiency going on. But if you're just running those nutritional testing, you're only looking at the end results of the actual disease state instead of actually looking at what the overall root cause is. So it's best to think about how do we optimize which labs to run and able to interpret properly to make our patients better, right? And if you go down the line to phospholipids, for instance, we talked about during a cell danger response, there's an increased saturation of phospholipids, which causes a decrease of fluidity going on. Normally, the cell membrane is very pliable, is able to communicate between cell to cell. The problem is, during a cell danger response, and then there's an innate immunity going on, well, it's in a mode to fight, right? It's trying to completely cave off any other dangers coming in and out. As a result, there's a decreased cell membrane. And it's so difficult to get these patients from point A to point B when there's such an influx of issue dealing with the fact that the cell membrane is completely locked. Or they actually have a lack of phospholipids as a reserve because of a constant inflammation that the patient might be dealing with, right? So I actually made a note about counterintuitive implication in dealing with the vitamin D, for instance, right? Such as mycotoxins blocking the VDR and chronic Lyme disease, causing a sequencing of progression of the disease itself. So there's no point of further pushing the patients and just giving them vitamin D. We need to measure and we need to look at what the overall potential cause can be, and we need to do a better job. So moving on to the phospholipid damage, we talked about how cell danger response and phospholipids coming into play, trying to lock that all in. One of the things is that cell membrane gets easily damaged by outside stressors. And cell membrane integrity gets locked in, we talked about before, and causes a poor cell communication going on. Namely, the goal of the entirety of that, and this is something that I encourage to do, and I have other physicians that are doing this as well, to allow the mitochondria to back away and getting into the overall healing response. By how? By applying membrane medicine and giving them phospholipids to allow the cell membrane to function and get this restoration done, but also it allows the overall inflammation that goes on and also prevents any of these shocking aspects the body has to go through. So in dealing with high toxicity and chronically ill patients, the overall cell membrane is in such a dysfunctional state that just giving them such binders or maybe certain antimicrobial agents out there, it may not do the overall trick because their overall CDR response is completely in a high influx state. You have to calm that down. By how? By utilizing phospholipids. So one of the things I recommend doing is using liposomal phospholipids and bioactive oils to allow to supporting the bi-layer of the phospholipid matrix. And the role of the phospholipids within the CDR, it's, when you talk about like gut health, for instance, has a mucosal protection by phosphocholine, namely, And then when you're utilizing these things, it allows the mucus to build up the actual surface to protect from other further damages. So I like using this a lot for patients dealing with SIBO issues or other gut inflammation that might be going on, especially Crohn's disease. And the proper phospholipid complex is essential as a building block for the CDR response to calm down. And this is something that I always give as a start, as a building block, and as a foundational support for patients to get better. And it explains very well in terms of the overall CDR model. And just as mentioned about before, the entire process of phospholipids and how that affects the entire saturation process, well, giving phospholipids would end up having to calm that entire process down. And then going to further down to the overall chart here, if you look at tryptophan, for instance, in an inflammation process, gut serotonin end up having to be affected, namely the serotonin receptor 4 is in such a higher overactivity and patients having higher anxiety issues, right? We see this and time again, patients dealing with gut inflammation, well, they have high anxiety issues or some type of mental health issues. And then when you talk about cholesterol, B6, and ceramides, for instance, these are all dictated by the mitochondria. So these are things that are very important. And then two things I want to touch upon, heavy metals as well. You know, I used to dispense a lot of chelation therapy using DMSA, for instance. It's not going to do the overall trick that you're looking for when you're doing capturing heavy metals. Why? Because the overall cell membrane, again, is completely locked, right? And then you're not able to detox overall heavy metals out of the cell. And so chelation-wise, it's not going to do the overall trick that you're looking for. And secondly, it also wipes away the overall lipid profile that the chelation is able to damage. At the same time, it also affects in terms of capturing all the electrolytes that's needed to support the electron transport chain or overall cell membrane. All right? So not surprising. So what I really like to do these days, and what I've seen being a lot more effective, is using phospholipids as a building block to allow cell membranes to get healed and then allowing other heavy binders. So one of the binders I love using is carboxy from Cellcore or using HMET binders from Cellcore as well. Those are two great binders to use to help deal with the heavy metal toxicity. And you got to support the drainage pathway as well, right? So those are things that I would end up having to add in. And then when you talk about gut microbiome, in an inflammatory state, food sensitivity occurs, right? And then dysbiosis and leading into the constipation and diarrhea aspect the patient dealing with. So talking about this right now, does it make sense to do food sensitivity? Many cases it doesn't. It can be a lot of waste of money that the patient has to spend. Why? Because we already know if the patient's having allergy issues or histamine response from certain food, we already know there's an ongoing issue with the mitochondrial dysfunction. So what do we need to do? We need to step back, assess it better, and then using other functional labs to look at the root cause issue and really helping to deal with that. And phospholipids, I mentioned before, could be one of them, really helping to support the detox pathway, right? And then using other antimicrobial agents to get rid of the pathogens or detoxing for that matter. So, in namely CDR, increasing CDR, all these disease states are interconnected one way or the other. So, again, what are we dealing with? Are we dealing with a chronic illness from just, plainly because of a person's genes? No, it's because of the fact that the cell is in such a danger response of fighting and wasting of the energy, resulting in a lot of these disease states. So, concluding-wise, the new understanding of the mitochondria is the brain of the cell. Okay, the genomics and the metabolic changes occur within the cell gets threatened, right? That's where the mitochondria come into play to detect that and allow the overall cell dangerous response to occur. And then certainly there's an abnormal pattern becomes fixated, right? The damage is done. You can't get the patient back through. So even in terms of the whole entire healing cycle, patient might be stuck in one or the other between CDR1 and CDR2, where there's an ongoing innate immunity going on, right? Namely, it could be where there's high inflammation process. So you have to look at this in a better way in dealing with chronic illnesses such as autoimmune. Treating metabolic shifts will not fix the mitochondrial back to healthy state. So we have to do a better job in managing the mitochondrial dysfunction by looking at the CDR and as a healing cycle and how we can incorporate that into our patient model here. So these are the references. And then one last thing I want to touch upon is my overall history. I actually had a heart attack back in 2015. So that's June of 2015. Up top, I was in the cardiac ICU unit for 3 days. And the picture below is only 3-day difference. I was in LA for a wedding. And then I was 33 years old. I'm 41 now. And June 2015, I thought I was just having simple severe heartburn. Actually, that wasn't the case at all. The pain got radiating down to my left arm. And like an idiot, I went to the urgent care center instead of going to the ER. And they said I had an unstable angina going on, that I have to be admitted to the ER. When I was there, the clinical presentation, I had no shortness of breath, no sweating, just complaint of chest pain. And I had troponin level less than 1. So in the clinical presentation right now, am I having an actual true heart attack? Probably not. When you look at a young guy like me, right, 33 years old, no previous other history, coming into the ER, looking at this. And the cardiologist thought I actually had pericarditis. After the echo, my left ventricle was not pumping correctly. Sent to the cath lab, 85% blockage in my left anterior descending artery. And then catheterization was put in, and I was in the ICU unit for 3 days. And then afterwards, I have this wonderful integrated physician that I've been working with since 2006. We're very good friends and colleagues. And she was questioning about whether I had some type of other infectious stressors that I might be going through that caused this, because there was no other previous history to dictate this. And then my visit to the hospital didn't end there. July 2015, I was sent back to the ER with a syncope. Look at the medication I was on. Isosorbide, beta blocker, valsartan, right? And on top of it, Brilinta. Even a regular patient ended up having to be an issue. A young patient like me couldn't even tolerate this. And then August 2015, I was finally tested positive for Bartonella through IGeneX. Mycoplasma pneumonia was positive, mold toxicity, and elevated Epstein-Barr. So basic diagnosis was an undiagnosed Bartonella infection may have contributed to the cardiac presentation. Right? So this was a tough moment because I didn't know what I had to do. A matter of fact that, am I going to have another heart attack from this? And the physician I was working with, she was smart enough and brilliant to looking at membrane stability. And I mentioned before, using phospholipids and PCs, that in itself saved my life. IV membrane stabilizing therapy was recommended, and along with that, using oral PC was really vital in resolving intravascular inflammation, dealing with biofilms and infectious burden. I ended up having to go through 28 total IV infusion, right? And then also utilize the sodium butyrate injection to deal with mold toxicity. And I continue oral therapy to decrease cytokine cascade. Now it's been 7 years right now. I've been Bartonella free, actually. It took me 2 years to clear this out, right? I didn't use any antibiotics. I'm not saying you don't have to. It's just that there's a different ways and modalities to fix the issue dealing with cell danger response and patients dealing with chronic infections like this, a hidden infection that I was not even aware of, right? And then during the whole lipid treatment itself, using PC, it causes your cells to detox, right? I mentioned about using PC to do heavy metal detox. What actually helps to detox overall other toxins as well. So it caused severe Huxon reaction. You know, I'll show a picture later, but I had severe acne all over my face and all over my body. So that was basically the body's way of trying to detox. And fatigue, headache, many cases I could only maybe work about 3 to 4 hours a day. Had to go home, couldn't function properly. I used heart rate variability to assess cardiac stress. And then one of the key things that really made a difference was working with a nutritionist to doing membrane stabilizing diet. Basically, it's a modified keto / paleo diet, right? by using bioactive oils to reduce carbohydrate intake and increasing cell membrane stability. Right? Some of the compound products I use, any prescribers out there, you might be very familiar with this particular list of compounds. Well, some of the things that I use, I don't particularly compound EDTA as much as anymore because, like I mentioned before, in terms of the different modalities in dealing with heavy metal toxicity. Nutritional support. At that point in time, I was not working with Cellcore, so obviously this protocol might have changed, but zeolite, some of the binders I ended up having to use. Liposomal PC capsules along with bioactive oils. Liposomal glutathione was one of them. Now, one of the things that I learned, at least having 7 years of clinical experience now in managing patients with this type of issues, understanding the use of proper binder is critical in patients' outcome and helping them to recover faster as well, right? And also opening up this drainage pathway is really important as well. If the liver drainage pathway is completely blocked, the patient might be having additional histamine response. We didn't know that as much, right? So that's really important in supporting that. Also, I did not really quite understand the influence of sympathetic and parasympathetic nervous system and how trauma, and especially emotional burden, comes into play in affecting my overall clinical outcome. And so nowadays, whenever I see patients and consulting patients in this type of scenario, I look at their overall history in dealing with trauma, right? A lot of patients dealing with mold toxicity, they have some type of trauma going on, either physical, mental, emotional stress, traumas that come into play. And it's very hard to get those patients back into a healing cycle unless you actually have to address that particular trauma the patient will be dealing with. So this before and after picture I posted on Instagram. And if you have Instagram, please follow me. I would like to follow you as well and connect with you afterwards. This entire picture was a 6-month difference, right? This actually fixed a lot of issues dealing with my gut dysbiosis. I used to have a lot of issues dealing with bad breath, and that completely resolved. Skin got cleared, right? Getting my life back, and was able to really clear the infection that a lot of people have trouble clearing out, which is Bartonella. So, I have to give a lot of thanks to God and the overall connections I have with different physicians I was working with, and then able to allow me to work with the patients that having similar issues like myself. So that basically is my conclusion of my talk, and I appreciate your time, and I have to thank Vibrant Wellness to allow me to be on stage and talk about this important topic of mitochondrial dysfunction.